Hello everyone. So in the last video I discussed till induction. So induction, what is induction, that you already know I guess by now, and inducer, responder, and how induction is actually playing an important role in the developmental fate of fate determination of cells. That I think you have now a good idea. So next I will move on to the types of induction. So types of induction is basically classified according to the to how the on the basis of the range of induction and on the basis of the choice of responder. So based on the range of induction, which means the distance between the inducer and the responder. So that is very important in case of induction. So for autocrine signaling, the name itself suggests that the cell is itself the inducer as well as the responder, because the receptors of the inducer molecules are present on the cell itself. So target sites are on the same cell. So there is no distance between inducer and responder. So this is known as autocrine signaling. Next is the juxtacrine signaling. So in this case, the inducer and the responder should be adjacent cells. Here you can see the inducer molecules are getting synthesized from this cell. So it is the inducer cell, and this is the responder cell because it has the target sites or the receptors for that particular inducer molecules. So it is there to respond to that inducer and act accordingly. So it should be adjacent to each other. Otherwise this signaling will not work. Next is the paracrine signaling. Here the distance between the inducer and the responder is increasing, but not more than a few cells. Like it can go up to across a few cells and then stop, but after that it will not, this signaling will not work. So target sites are on adjacent cells, and there is rapid breakdown of ligand. So here you can see the ligand was already connected to the cell, and here the receptor was also connected to the cell membrane. Therefore there was a direct connection between the inducer and the responder. But in case of paracrine signaling, there is a transport of the inducer molecules up to a few cell distance, and then it gets bind to the target sites present on the receptor cells. So next is endocrine signaling. So here in paracrine signaling, you can see there is no involvement of blood vessels. The inducer molecules by diffusion by in the extracellular space, it can transport freely. But in case of endocrine signaling, the target sites are so distantly located from the inducer molecules or inducer cells that it has to be transported through the blood vessel in the bloodstream, and then it reaches the responder cells. So these are the four kinds of signaling or the four types of induction based on the distance between the inducer and the responder. Next is on the basis of the choice of responder. So there is an inducer and a responder, but how the responder will react to it or how it will respond to it, the induction signal, that is the choice of the responder. So according to that, the induction can be classified into permissive and instructive. Permissive induction is basically the cell is already determined to a particular fate in a particular pathway, so it does not actually require an instruction to act accordingly. Instead, in a presence of a suitable environment, this cell will automatically differentiate because it already has all the signals or all the necessary necessary molecules required to differentiate in this kind of fate. So you can see the differentiation is occurring just because it has all the necessary factors available. It's in a suitable environment, and there is no such instructive signal given. Because of that, the differentiation is happening. There is nothing like that. But also, in absence of these factors and in absence of a suitable environment, this differentiation will not happen. But in case of instructive induction, the instructive signal is very much essential for the differentiation to occur. Now, what is this instructive signal? If this signal is absent, then the cell will form something else. It will move on to an alternative developmental pathway. It will not follow the pathway which it is usually supposed to follow. So here is the difference between permissive and instructive induction. In case of instructive induction, an instruction or signal is very important, and because of that the cell can follow either this path or an alternative path. If the signal is absent, it will go on to the alternative path. If the signal is present, it will move on with the traditional pathway that it is supposed to follow. But in case of permissive induction, there is no such signal, and the cell will automatically differentiate into a particular cell type which it is supposed to form after differentiation. And in absence of these suitable environment and all the factors, the cell will not differentiate at all. So under instructive induction, there are two kinds, appositional and morphogen gradient. So appositional is basically when the inducer is brought nearby to the responder cells. So you can see the green is suppose the green is an inducer cell. So wherever the inducer cell is in close contact with the responder cell, you can see that part has differentiated differently and the sides are of different color, which means they are forming something else. So appositional means based on the position of the inducer cells, the responder will be given an instruction or a signal, and because of that there will be differential fates or differential pathways. Where the position is different, then those cells will form something else, and where the position is quite adjacent to it, it will receive the signal and it will differentiate into something else. Next is the morphogen gradient. So here you have to understand what is a morphogen. Morphogen is basically a signaling molecule that spreads from a localized area, and it gets distributed according to a concentration gradient. So according to the concentration gradient, different portions of the cell or the cytoplasm will form something else, and based on that, as you can see here, if the violet color is a morphogen, then it is spreading from this side to this side of the cell layer. So what will happen is that the cells of this layer side will form a different organ, and the cells of this side where the least concentration of the morphogen is present, it will form something else. Like I have already explained about bicoid and nanos when I was doing the syncytial developmental pathway. I already explained how the different concentrations of bicoid and nanos determine which portion will form the head, which portion will form the thorax, and which portion will form the tail. So next I will move on to competence. So this is another very important term when you are learning about induction. So competence is basically, suppose the inducer is giving signals continuously, but what if the responder is not able to receive the signal and also not able to know what exactly you are supposed to do with the signal? Am I supposed to go into this pathway or this pathway? What does this signal mean? So how to interpret the signal, how to receive the signal properly and act in an appropriate manner. So this is very important. So this ability of the responder cells is known as competence. So whenever a cell loses its competence, that means the inductive signal will not have any effect on that particular cell, and therefore that cell will lose its fate, like it will stop being differentiated further. So it will not move into any developmental pathway, and therefore the cells become committed. Whatever the cell was at that point of time, it will be that till eternity. So it will not receive any signal further. So you can see what is loss of competence mean. Loss of competence means the loss of cell surface receptors. Like I showed you, the inductive signals require a receptor or a target site where it will get bind to. So if the cell loses all its receptors for that signal, then it will never receive the signal and also never act according to what the signal means. Or if the cell loses its transduction apparatus. Transduction apparatus is nothing but how the signal is transduced inside the cell, and by a downstream pathway. You must be knowing that several signaling pathways have downstream molecules to which the signal is getting transmitted. So if that apparatus is not present, then the cell will lose its competence. Also, if the downstream target transcription factors, like the transcription factors which are responsible to activate the transcription of target genes, they are not present, then also the cell will lose competence. So the action of the signal will not be completed. There will be no outcome of receiving the signal. So that means there is a loss of competence.